Welcome back to the Business of Biotech. I'm Matt Pillar, and I'm likely to get way out of my depth with today's guest in a big hurry, so it should be a fun listen. She's Chief Business Officer of a new company that's a bit of a departure for us. The company's not, at least not right now, developing a pipeline, but rather it's leveraging generative AI to do all sorts of useful things like reading and writing therapeutic proteins from scratch and making accessible to the biotech community an open sourced AI generated gene editor called OpenCRISPR. On today's show, ProFluent's Dr. Hilary Eaton is with me. And while I'm not going to let her take me too far into the AI rabbit hole, we are going to learn a bit about how it works and, importantly, where and when it doesn't work, its applications in biotech, why ProFluent is doing what it's doing, much of it for free, I might add, and what's inspired Hilary's life science career path, both personally and professionally. Hilary, welcome to the Business of Biotech.
Thanks so much for having me. It's a pleasure to be here.
I'm super excited that you're here to join us. I had the benefit of a conversation with you. For our listeners, just so you know, Hilary and I talked a couple of weeks ago, and I've been pumped to do this interview since we chatted because I was so excited and inspired by Hilary's enthusiasm. I'm setting the bar very high from the outset here, Hilary. You got to bring that. You got to bring your A game. But I want to start out with your a little bit about where you came from and what got you into the space in the first place. So a little bit of background. Hilary earned her Ph.D. in molecular cancer biology from Duke. She did her postdoc at Harvard. And then, Hilary, you got seemingly, like looking at it on paper, you got directly into licensing and business development deals. So that's where my curiosity gets piqued a little bit. What motivated this, what looked like a scientific or therapeutic sort of development-driven path? What motivated that switch early on into licensing and business development?
Yes, no, that's a good question. And I think, the story is that it's always more messy behind the scenes than it is on paper. I am a bit of a poster child for don't know what I want to be when I grow up, and maybe still don't know. When I think back, to the extent I remember it, to my decision to go to graduate school, it really comes back to science was in my family. So I grew up watching my dad. He was a career scientist, a chemist at Pfizer for his entire life. And he headed the team that developed Lipitor, their famous cholesterol lowering drug.
Yeah.
And I somehow got the, very mistaken idea that all scientists just work on drugs that millions of people take. And that sounded great to me, right? That sounded like a really direct path of helping people. And as a side note, I think now that I've become a parent myself, this really does make me think carefully about sharing both my successes, but also my failures with my kids so that they don't get sort of like a skewed view that everything in life works out. And in fact, when I had a follow-up conversation with my dad, I learned a lot about the many projects he worked on that were not Lipitor, that did not make it to the clinic. And it was a very enlightening conversation about, the ups and downs of a career that on paper can look like one very major success.
Yeah. That's pretty cool. I mean, that's really good perspective,
Yeah,
like I
yeah.
going through that with my kids right now. My daughter's about to go into college. My son's going into a second year of college, and they're, in that formative stage, and their perception, you don't really often think about what their perception of what you do and how you do it how that might influence them. Like you just looked at dad as like the Lipitor making hero, right?
Yes. Lipitor making, Lipitor taking. He takes his own medicine.
Yeah.
And so I kind of, it was sort of just a given that I was going to go to graduate school and sort of follow in his footsteps. I mean, he was always so inspiring to me. But then sort of like back to the career pivot. So, maybe halfway through my PhD, and that sort of like you've gotten through the prelim, you're now working in a lab. And I started realizing that a lot of the basic science projects that are carried out in academic labs are never going to directly affect a patient's life. They're often very crucial steps to understanding the underlying mechanisms or pathways that are affected by disease. But they're many steps away from a medicine that's actually going to affect lives. And so during my postdoc at Harvard, I started to search for a more direct way to help people. And I'm a big fan of thinking about sort of inflection points
Mm.
and also pondering on how sometimes what can be a pivotal point for you, the other person doesn't even realize that they said something that affected you. And I come back to there was a lunch and learn seminar with the then chief scientific officer of Dana-Farber Cancer Institute. His name was Barrett Rollins. And he probably doesn't even remember this, but again, it made a huge impression on me at the time. And he was asked by somebody, how did he become CSO? Like, what was the transition from being a PI, being a lab head, over to a more administrative managerial role? And
Mhm.
in the course of his answer to that question, he mentioned what, was in principle a fairly simple exercise. It sounded like something that like a career counselor would give you to do back in high school. And he said, make a Venn diagram. And on the one side, have it be the things that you're good at versus the things that you're not, and be honest.
Mhm.
And then have the other side be a list of things you enjoy doing versus the types of things that you simply tolerate or maybe in fact hate. Um, and then he said, if you in your current role are not at that intersection between things you are good at and things you like doing, that does not mean that you are a failure at the thing you are trying to be. It means that your skill set and maybe your enthusiasm would probably be better fit for a different role.
Mhm.
Um, and that really struck me. And so I went home that night and made a Venn diagram. And first of all, it was a lot harder than I thought. Like getting honest with yourself about stuff that you aren't good at or that
Yeah.
you don't like doing, that can sometimes be a little depressing too, to see, kind of look at the current job you're doing and the list of things you don't enjoy is much bigger than the list of things you do enjoy.
Yeah.
Um, but it gave me a hint about what to look for next, and that was exploring the business side of science, being able to still leverage the skills I'd learned throughout graduate school and my postdoc, but sort of applying, some of my strengths in communication and in problem solving towards how could you make medicines and get them successfully to patients. And so I started sort of on, you could say, the light side of the dark side by dipping my toes into technology transfer at Dana-Farber. And that was really a crash course in many things that I had expected. I got to learn about agreement negotiation, intellectual property, budgeting, alliance management, all things that I had never touched on as part of my scientific training. But surprisingly, I also got a crash course in expanding my scientific horizons.
Mm.
Um, you work on these niche projects during your scientific training, and so much interesting science goes on beyond your project or your department at a research institute. And so I got, honestly, a broader picture, I think, of the science being done at Dana-Farber during my time in tech transfer than I had as a postdoc. With my head down working at the bench. And it was, I found myself every night back on PubMed, trying to learn about new areas of science that I hadn't been trained in. Like there was this new field called immuno-oncology. I knew
Mhm.
nothing about it, but I got assigned a portfolio for a couple of lab heads that worked in that space. Um, and it was absolutely fascinating to me to get to see kind of the best things, the most cutting edge things that were going on, and then to try and figure out which one of those things had legs to become a company or to be licensed to a pharma to become a medicine. And that was my introduction to business development.
That's wild. When you, after you had that exposure and a little bit of that experience, um, it, how did that reshuffle the Venn diagram? Deck's a bad analogy. How did reshuffle the Venn diagram? Like if you were to do that Venn diagram over again, did you find like, oh, the business side of things is kind of falling in a different place? Maybe the balance is a little bit different, or was it? I'm just curious about like how that
Yep.
that experience influenced.
I think, no job is ever perfect, but I found that the balance skewed a lot more towards that sweet spot of things that I hope that I'm good at and things that I really fundamentally enjoy doing. It's
Yeah.
whenever I give career advice to folks, in my network, it's that, indescribable feeling, or it's that like when I look at people, you can see when there's a sparkle in their eye. You can see when they're enjoying what they're doing in that moment. And you see times when that spark kind of dims and it's just the rote grind. And I don't know if there's ever going to be a job where your eyes are sparkling 100% of the time, because that might
Mhm.
just be complete burnout or just, unrealistic. But I definitely noticed that there were a lot of things that brought that spark to my eye. This idea of, um, doing, for example, a deal negotiation. Ultimately, it's framed as you're on opposite sides of the table, right? Yeah. And trying to get the best deal for your side of the table. But that's not really what it's all about. What you're trying to do is get at whatever science, whatever drug there is to be made there. And both of you want that. And each of you have some very real concerns about things that could go awry on the way to building that drug. And so figuring out sort of what the actual concerns are under the surface and then finding creative ways to compromise about those, that to me is fun.
Yeah.
It's a fun puzzle to work out.
Yeah, that's pretty cool. And I think the Venn diagram would probably help a lot of people because like I've had conversations with people who have your academic background who would decidedly say that that is not fun, Hilary, not fun at all, you know? So those exercises, I think it's good, like as your career goes on. And speaking of deal making, I think next stop in your career, you're doing big deals at Editas, right? Was that next after Dana-Farber?
Editas was next. Yeah.
Yeah. And during that time, you shared with me, and you said we could talk about this. It's personal, but you said we could talk about this. During that time, when you're doing these big deals for Editas, your career's starting to take off. You find out that you have, in fact, a personal connection to rare genetic disease, a very, very personal connection. So tell us a little bit about that story.
I'm happy to actually. I think, honestly, if more people are able to be vulnerable and talk about their own experiences, it can sometimes help all of those folks out there who are living through similar things but feel so isolated because some of these things don't get talked about. So, as I was looking at the Editas role, in the background, my husband and I had been trying to have kids for some years without any success, and we ended up doing IVF. And so, ironically, I was actually waking up from having my eggs harvested when I got the call from Deb Palestrand at Editas saying that she was offering me the job. And I just had a nurse who said, okay, you've had general anesthesia, no big decisions, no driving. And then I got that call and I was like, yeah, I'm making this decision. Um, and so, while I was at Editas, had two little IVF miracle babies, and was just so grateful that the career that I had embarked upon and science was able to sort of make a dream come true for us.
Yeah.
And then over the course, it's so busy, like two parents working full time in the biotech, two young babies, and it's just kind of like all a blur. But at some point along the way, we kind of experienced one of those like worst moments that you can have as a parent, where all of a sudden you realize that your kid is missing developmental milestones.
Mhm.
And like, when it's your first kid, you don't really have much to
Benchmark.
compare it to, right? You don't have benchmarks out there. And so it's kind of like, yeah, she's taken a while to sit up, but like, I don't know, there's a range. The pediatrician every time tells you, oh, kids are a range. It's a whole spectrum, right?
Yeah.
But it was more prominent in our second daughter. And at this point, actually, I think I had transitioned to Bio. And I she had some hearing loss at birth, but it's so hard with babies to figure out how profound certain things are. They don't exactly cooperate on the exam table with various tests. And so it wasn't until she was 9 or 10 months that we really realized how significant the hearing loss was. And there were a variety of other symptoms that kept popping up. But the biggest thing is, we were in it at one of the hearing sessions, and somebody says, oh, could she sit by herself in the chair without you holding her? And I said, oh, no, she can't sit up by herself. And they said, really? She's 10 months now. That's, she should be able to sit. And so
Yeah.
that led to being introduced to early intervention, which the state of Massachusetts does when you have a child with two or more developmental delays, and a whole host of additional specialists to look into why she was missing these gross motor and these fine motor milestones. And, what it came down to in the end is that she was so hypermobile, so flexible that it was hard for her to build strength and figure out how to do some of these crucial things like sitting up or pulling to stand or crawling. And I can remember a point where I was in I think it was an orthopedic office. And Olivia at that point was, I think, maybe around 26 months
Mm-hmm.
and not walking. So significantly delayed
Mm-hmm.
and just feeling extremely frustrated because we had gone through sort of a laundry list of different, pediatric specialties and nobody was giving us answers. And sitting in this office and the doctor turns and looks at me and says, Mom, are you bendy? And I said, I mean, yes. What does this have to do with this conversation? She was like, do you have party tricks? Like, are you double jointed? Can you bend joints backwards? And I said, yeah. And she was like, I know what's going on here.
Yeah.
She's like, there's this thing, there's these host of connective tissue disorders where the most obvious symptom is this hypermobility. And she's like, and I think what we've got going on here is that this little girl, is just so bendy
Mm-hmm.
without the corresponding strength, because how would she have built that up?
Right.
Right. I think that she needs, some braces. She needs some braces on her ankles, and just stabilizing some of those joints is going to really help with the immediate symptoms. Um, and it was just that moment of, could this really be it? Do we
Mm-hmm.
have some light dawning? And so they fitted her with some custom orthotics. And we kind of thought, yeah, try sticking your 2-year-old in ankle braces. There's no way she'll take it. This is going to be a grand tantrum. And instead, we had that moment in the office where they put the ankle braces on her and she turned and looked at me. And then she pulled up to stand. Like,
Wow.
Mom, I was just waiting for this.
Yeah. A little support. Yeah.
Yeah. And she was walking within like 2 weeks after that. And so, you know, getting a diagnosis for a genetic disease is often a really long and discouraging process, because there are so many of them, and some of
Mm-hmm.
them come with such, amorphous constellation of symptoms. And so I think, we got phenomenally lucky to be in Boston and to have some amazing physicians in our network that were able to get us there, but one of the issues here is that there aren't, just as there aren't treatments, there are also not diagnostics for many of these things. So
Yeah.
the this particular disorder is called hypermobile Ehlers-Danlos syndrome, or HEDS for short. And for this particular subvariant of disease, there are no known genetic variants. So it's not, you can't just send a blood sample in and have a doctor say, yes, you have the mutation, or no, you don't. Instead, they're looking down sort of, a bucket list of clinical symptoms. And if you meet enough of the criteria, then you're deemed to have the disease. And the thing is, my girls at the time, 2 and 4, they were too young to have had some of the life experiences that would have led to some of the symptoms, like repeated dislocations,
Mm-hmm.
slow wound healing. And so the doctors came to me and said, Mom, an alternative approach here would be, if you got officially diagnosed, that would allow us to then diagnose your children. And so I found myself in a doctor's office at, I don't know, 37 or something, being asked all these questions. And all of a sudden, so many different symptoms or weird medical things that had happened to me over the course of 37 years were drawn together
Yeah.
into a diagnosis. And I still can remember, the doctor at the near the end, the geneticist at the end of the appointment said, heartwarming that you're doing this, in order to get your kids, diagnosed and on the path, to ameliorating their symptoms. He was like, also, you've just been living with this for several decades. You're kind of screwed up health wise, and you need doctors.
Yeah.
You need to go see specialists for some of these different conditions. And I just thought it was too funny, that progression sort of like to diagnosis,
Yeah.
that like symptoms that I had experienced somehow weren't worth it to me, or didn't rise above the radar to, maybe I should go see a doctor about this. But when it was my kids, the mama bear comes out and you're like, okay, let's get this solved. Right.
Yeah.
So yeah, that was the path to getting diagnosed.
You didn't call your mom and say, "Hey, thanks, mom, for not noticing that it's gonna be 36 months to sit up in my high chair."
Well, the funny thing was, I had normal progression with motor development. What they're finding with a lot of these disorders is that you see something that's called anticipation with generations. So maybe grandma has it, mom
Mm-hmm.
has it a little worse, kid has it a little bit worse. And so it actually made a lot of sense. It's actually led to our whole extended family being tested, because, there's the part of this journey that's getting an answer for yourself. there's also a part of this journey that's how can you help the broader community? So when somebody shows up in a clinic and there's no family history of any of these symptoms, it's very hard then to look for what in their DNA might be causing these symptoms.
Sure.
When you have a large extended family with folks who are quote unquote normal and folks who are less normal, you can start finding patterns where you can say, okay, maybe we have 75,000 SNPs or random mutations in our genome, but there's only this one SNP that's in the affected females in this family. So we saw it as both answers for ourselves, but just as science, my husband and I are both scientists. My dad's a scientist. We had this, immense curiosity about what is causing this and can we add to the knowledge in the field?
Were you already thinking about the impact of data sets in science and in life sciences and therapeutic development when you experienced this, or did it sort of inform your perspective on data sets? Boy, data sets are really important to therapeutic development.
It's a little bit of both. So, I mean, during my time at Dana-Farber, one of the things that came up is that, they treat so many patients every year. And there's data from treatment outcomes, there's data on symptoms, there's also data in the form of actual patient samples, like biospecimens. And you learn a lot from seeing how somebody's tissue changes over the course of disease progression and then treatment. And so one of the things that we worked on at Dana-Farber is making sure that when you're writing up IRB consents, IRB protocols, to enroll patients in clinical trials, that you can really explain to them how valuable their data is and how it might be used in the development of new medicines and what precautions you take to keep the right privacy around that data. And then to find myself sitting on the patient side of that seat, reading patient consents that I might have had a hand in writing, it hits hard. And there can be that instinct of like, no, my genes are mine. My information is my own private. And I respect people very much who make that decision. I think maybe, somewhat uniquely because of the science background, part of me said, yeah, I'll put it all on the table. If somebody can learn something from my cells, my DNA, that means there's a mom down the line whose baby is diagnosed at birth and who learns some of these, developmental things on a regular schedule, I will have contributed in a meaningful way that has, nothing to do with like a personal achievement at all. It's just something that's like inherent in my body.
Yeah. Yeah. All right. Before I ask you the next question, how are your girls doing today? How are they doing now?
They are doing phenomenally. So, one of the things that we've really tried to do is treat the medical world. It takes a village, let's put it that way. And we've assembled together a team of specialists that track throughout. How do I put this? You have connective tissue in literally every place in your body. So there are a whole range of things that can go wrong with this disease. And it used to be that they would only pick up patients fairly late in life. I think the last quote that I heard from the patient community is that the average time to diagnosis for Ehlers-Danlos is 14 years. So you're often dealing with folks in sort of young to mid-adulthood. Now Boston Children's Hospital is seeing a lot of much younger kids coming in.
Yeah.
And they don't really know what disease progression is going to look like. They don't know how early certain symptoms are going to pop up. And so, we just made sure that we got, the right team in place with regular check-in schedules. And it's, thing will solve problems. It can be a little bit like whack-a-mole because you think you've gotten to a good state and then something new pops up. But I think I'm just so incredibly lucky that I have this network of friends and colleagues and former colleagues that I can draw on when some of these new things pop up to figure out, how to frame the approach. It's, something happens, so figure out how to solve it, figure out who the people are to talk to. And they're doing wonderfully. They're both in school now. They're just really thriving. And in some ways, as much as I was, sad to find out that I have a disease that I had passed on to them, my therapist, my executive coach, all my friends, the point to drive home is that who better to parent a child with rare disease than a mom who already knows what it's like to live. Like, there's nobody better to advocate for them in some.
Yeah, and to demonstrate the strength that's possible in spite of the disease, right?
100%, 100%, because that is one aspect of my persona, that I have a rare disease. That does not define me.
Mhm.
Does it limit me? Perhaps in certain ways, but it's interesting to figure out how you can sort of like find hacks or band-aids or approaches that allow you uh, to ambitiously go after your career goals while still dealing with some of these things that are happening in the background. Sometimes I almost think that maybe that in and of itself is what led to me having certain strengths in my career in business development, because I'm not afraid to go out there and advocate for things and ask the hard questions and try to come up with creative solutions.
Hmm. Yeah. Well, I hope you're still buying an annual bottle of wine for that pediatrician who asked you if you're bendy, because thank God for her. That's fantastic.
Absolutely.
Yeah. We need to get back on the career trajectory here,
Yeah.
right? You said you were the move from Editas to Vor. So that was Vor and Tome, I think, were the next two stops
Yes.
on your journey. Tell us a little bit about why you made those switches.
Yeah. And so it's interesting. I've definitely had some mentors along the way who've advised me to branch out or to generalize. I kind of, when I took the job at Editas, I honestly didn't even understand what CRISPR gene editing was. It
Hmm.
was, a technology in its infancy. I had a couple of postdoc friends who were using it for screening, but I had no idea that it was going to become sort of a next frontier in genetic medicines. Um, and as I looked, at beyond Editas, I again, these mentors were saying, okay, you've had, an incredibly successful stint at, one of the pioneering editing companies. Do something else to, go into a different area of technology or a different area of disease. But for me, when I looked at the field, there is so many good science. There's so many other good things, particularly around the Boston area, that you could go for. But I do
Yeah.
not see any other path to alleviating or even curing rare genetic disease that's more promising than gene editing in its many forms.
Hmm.
And so I've stayed put. That's definitely been a theme for me, staying at companies that are exploring with different generations of gene editing systems.
Yeah.
Um, and and in many ways, it kind of feels like that, is the perfect intersection of life experiences for me. That, I'm trained as a scientist, and fundamentally, gene editing is not an easy or a simple process. It is, a very intricate system, complex biology. And in my job as in business development, I really never know who I'm going to have to explain or interpret to. I could be dealing with a Nobel Prize winning scientist who wants all the details, or I could be dealing with somebody who has more of a finance background and needs it to be, explained in a simpler manner, that
Hmm.
they'll be able to deal with. And so, you've got that part of it. And then on the deal negotiation side, one of the things that's been really interesting then is getting to see, as this, field of gene editing has started blossoming over the last decade or so, seeing what it takes to have success or not in the field of biotech startups, seeing, that combination of good people and good ideas and good strategy, and how some of those things pan out, like what decisions you make with the data you have in hand at the time versus what decisions, you might have made if you'd had 2020 hindsight.
Hmm.
And then finally, that patient perspective, right? It is, I remember the clinical indication selection exercise at Editas, and it's very patient driven, it's very patient focused, but that patient is also other. That patient is sort of, cut and dry facts that are on a piece of paper. What does the disease progression look like? What are the symptoms? What's the underlying genetic mutation? Can our technology fix that mutation? And once you get a diagnosis, the psychology of that exercise is so very different, because it becomes, that could be my daughter,
Yeah.
right? How are
Right.
we approaching this safely? Is this, can we, with the way that clinical trials need to be set up, are we going to be able to intervene early enough in that child's life to have meaningful clinical benefit? It just, it changes the equation. And so I think, one of the things, go
The
ahead.
equation and a sense of like does it up your sense of urgency?
Definitely ups the sense of urgency,
Yeah.
of impact. And I've seen folks out there on LinkedIn and X, talk about the explosion of gene editing companies, and do we really need that many? But I would argue yes.
Yeah.
There can't be too many companies, right? There are so many rare diseases and so many patients out there who are looking for answers that I really don't think there can be. And it also then has changed in certain ways the way I view the business of medicine, right? It's phenomenal to see, like the Casgevy, the first approved medicine, CRISPR medicine for sickle cell that CRISPR Therapeutics and Vertex, brought to the clinic.
Mhm.
Seeing folks get cured and no longer having these horrible vaso-occlusive episodes with, pain and blocked blood vessels. That is amazing to see. And those drugs are priced accordingly. I mean, hundreds of millions of dollars of research go into, every one successful drug has 99 failures sitting in the background.
Mhm.
And yet when you look around the world, the patient population of folks with rare disease is not limited to, rich people in developed countries that have great insurance. It
Yeah.
spans the gamut. Nobody's spared, right? And so you start thinking about, we are very early in the arc of a new technology. Things are usually most expensive at the beginning because they're so new. And you see, as things become more common and commoditized, you do see prices come down. But it really did start get me thinking about like, how do we Jennifer Doudna and her Innovative Genome Institute, right? This idea of democratization. How do
Mhm.
you take such a powerful technology that has applications that go way beyond human therapeutics? When you think about sustainable crops, climate change, and how do you responsibly use it for some of these things? How do you use it for the betterment of society, and how do you do it in a way that's approachable for as many patients as possible?
You're doing a beautiful job. God, you're a great storyteller. Fantastic storyteller.
Thanks.
Uh, and you're doing a great job setting up my segue to the next career pivot. Um, I talk to a lot of physician scientists turned biotech founders, and they tell me, well, I was administering the thing and the things, and then I made the decision to get into biotech or biopharma because I wanted to make the thing, right, that I was administering. So here you are, you're working for companies that are making the things, a few of them in a row, and doing well, I'll add. Great business development career with these companies, doing what a lot of people aspire to do, making the thing, developing the thing. And then last year, early this year, you decide, now I want to go do something else. And it's not with a company who's making a thing per se in the context of, in terms of a therapeutic. Now, like I said, I think you were sort of leading into that question when you were talking about these bigger problems, these bigger challenges that your current company, ProFluent, is attempting to play a key piece in the puzzle, right, of solving. So tell us about that. What prompted and how was what was going on in your mind? What did you struggle with when you said, you know what, like I think I'm going to move away from working in BD for therapeutic development companies in the gene editing space, and I'm gonna jump on this. I won't
AI.
say, yeah, AI, yeah, and I was gonna,
bandwagon.
yeah, I don't want to call it a I mean, yeah, I'm fully on board that bandwagon, and I don't even want to call it nascent. Like it's here, right? It's happening. So, but it's still big transition for you, big decision, right? Walk away from therapeutics development and take on a risky role with a startup company that doesn't really have a product per se when you joined. Like, what was going on there?
Yes. And so, I mean, as you said, I was drawn to the previous companies, to Editas, to Vor and Tome, because they were all, pioneering incredible editing technologies and turning those into medicines for rare disease, for immune disorders, for cancer. Um, and as I experienced both those specific company builds, but also sort of getting the bird's eye view of the developing gene editing field as a whole, one of the things that I was struck by is a pattern of needing a new approach to protein engineering. So why would we need a new approach? What wasn't working or what isn't working? So in many cases, CRISPR being one of them, the starting point for a therapeutic is a protein sequence that has essentially been copied and pasted from nature. And often it's by random chance or discovery. It's
Mm.
not that somebody's trying to find that particular thing in nature. It's that somebody's working in bacteria and then discovers this thing that does something, and they start thinking, huh, how could that be applied? Could that be applied to human medicine? Um, Now, the tricky thing is that a protein that evolved in nature to carry out a certain function might not be the perfectly designed solution for a given human use case that you're looking for. And so, if we want to adjust the function, how do we go about doing that? We need to tweak it slightly. How do we go about doing that? And so there are sort of, different approaches or different eras of protein engineering. It's not a new thing. It's been going on for decades, right? So
Mm.
one is sort of the manual route where you could carry out random mutagenesis or directed evolution, and trying to guide the protein towards those criteria you're looking for. This is incredibly time and resource intensive. It often is sort of, again, random mutagenesis does not really imply that you have much of a hypothesis. It's your sort of like randomly tinkering with different areas of the protein to see if you then get results that you want. Um, and often what you end up getting is incremental improvements at best. More often you break the protein
Mm.
because you tinker with a fundamental area of it that's necessary for function. And so, then maybe moving on to the next era or sort of a different approach, we've got folks who do metagenomic mining. What does that mean? Well, they look at, I lifted this particular sequence from this particular organism. It's almost right, but it doesn't do these other things we want it to. So I just need to broaden my search. I need to look at more organisms. And in the diversity of organisms, eventually I'll find something that does have my list of criteria.
Mm.
And certainly some folks have had success, looking into, volcanoes and ice flows for more genetic diversity. But I would still argue that that is a fairly needle in a haystack approach. And it often results in functional trade-offs. And so what do I mean by that? So when you look at natural evolutionary processes, often the selection pressure that's being put that leads to sequence changes is focused on one particular attribute of that protein, of that enzyme. So for example, perhaps the host organism is small. So the enzyme itself needs to be smaller. It just isn't going to fit. It's not going to be able to do its job.
Mm.
But in the process of evolving and achieving that smaller size, it's possible, I'd argue even probable, that other aspects of that protein's function are going to be altered. And so change is kind of like a double-edged sword in this scenario. It's, you achieve one goal, but you sacrifice other features that weren't critical for fixing the current problem in that original organism or
Yeah.
survival of that organism, but they are key for the therapeutic problem that you want to solve. And so, in comes ProFluent and generative AI, which I'll be frank, I knew nothing about. I'm a huge sci-fi aficionado, but I knew nothing about real life AI before being introduced to ProFluent. And, you know, one of my first thoughts is this is the next era of protein design, where we're moving away from sort of accidental discovery or sort of, tinkering around in a manual way. And we're really moving to intentional, bespoke design aided by artificial intelligence. And, they have these large language models. And those large language models, they're not limited by natural sequences. They don't involve manual labor. There's no functional trade-off. It's when you look at the combinatorial space of a single protein, it's greater than the number of atoms in the world. And so there's no way that even a team of the best scientists can sit down and explore all the possible sequences, right? But generative AI can do that in a matter of minutes,
Mhm.
right? It helps you narrow the funnel of what could be possible, places evolution hasn't had a chance to explore, and pull the gems out. And so rather than sort of starting with a molecule and then trying to kind of like jam it into a therapeutic use case that it may not be quite perfect for, but it's sort of good enough, we're really trying to kind of flip that paradigm for drug development upside down. Start with the problem. What is the problem you're trying to fix? And then if you had to imagine the perfect drug that would fix exactly that problem, what characteristics would it have?
Mm-hmm.
And once you have that list of criteria that you think define the ideal system, then we task the AI with building exactly that thing. And that just blew my mind that could that even work?
Yeah.
Right.
I'm going to bounce around here with my questions because I'm going to go with like there's primacy and recency. Ask questions about something you said earlier. I'm going to go with recency.
Okay.
This description that you just illustrated, start with the problem and then ask what are the attributes of the solution to that problem, and then go out there and use the data. It seems to me in that situation, the data, at least at this point in time, might be the weak link.
Or the strong link, depending
Or
on
where, yeah. So let's talk about that. Like I said, I'll probably bounce around here for a minute, because I'm already in over my head. But hey, here we are.
here we are.
Where does the data come from? Like, where does the data come from? And like in that scenario, do we have? Do you think enough data to make impact, like to query, to apply these models and make impact?
Yes. So I will caveat by saying that I am not too unlike you. I am definitely in over my head. And anything I know about AI, I have learned from my team members, but especially from our
My
investor.
my listeners are calling your bluff right now. They've been hearing from you for the past 45 minutes, and they're like, this woman is not anywhere near over her head.
I'm trying to, it's definitely sort of like a fire hose, right? At the beginning of a new job, picking stuff up. And we're lucky enough, we have a board member named Fraser Kelton, who was the former head of product at OpenAI for ChatGPT and DALL-E. And then ProFluent's own CEO and co-founder, Ali Madani, who was one of the leading researchers at Salesforce that did all of this foundational work on applying large language models for protein generation. And so, they've definitely helped bring things down to my level in terms of understanding, what are large language models and how did it go from an area? If you look back 5, 10 years ago, your iPhone model, I don't know, 2, 3, couldn't finish a sentence for you. Like its predictions, right? Were often hilarious. I would sometimes just like take the prediction because it was so hilariously wrong, right?
Yeah.
So how did we get from that world to a point where we have something like ChatGPT, where you can say, write me an essay in Shakespearean style on soccer, and it needs to be 2 pages long, and it can just do that, right? That it seems like an incredible advancement. How did we get there? And as they describe it, the two key ingredients, one really is fundamentally advancements in AI. There's something called the transformer algorithm that apparently had a profound effect on models' ability to learn and generate useful results. But the second key ingredient is what you are alluding to. It's data. It's the scale of data. It's the quality of the data. It's the bias or lack of bias in the data. And so, when you think of, how things like ChatGPT were able to have success, there are millions, if not billions of examples of text floating around on the internet in, books, all these different things, right? And so that's a rich abundance for the model to scrape and learn from and learn, just by like, when you think of a young child learning language, right? Some of it's intentional teaching, but a lot of it's just passive, absorption. They hear things, they learn grammar unconsciously, and that's what these models do. And so now if we turn to the world of protein discovery, that same abundance is mirrored in nature. We have, the world has provided us with this abundance of natural protein sequences over the course of evolution.
Yeah.
And much of that data is just like readily available. It's just out there. It's public.
It's I guess that's part of my question though. Is it analog? Like, is that data that's out there in nature? Like, if I think the publishing industry certainly was an early to the internet, early to the cloud industry. I haven't been around the life sciences space long enough to know that they were jumping on that bandwagon from the outset and saying all the data, all of it needs to go. It needs to be digitized and it needs to become accessible. So that I think that's kind of central to my question.
Yeah. So I would say that there are ways in which some data doesn't exist. And And we can get into that, I think, later, maybe when we talk about sort of like negative data. When
Yeah.
it comes to things that are natural sequences, as opposed to, a drug that somebody's trying to develop, when we think about what exists in natural sequences, yes, there is so much data out there that's digitized. Our current frontier model is being trained on over 80 billion protein sequences
Mm.
that are just out there. I can't say that they're often uh, perfectly curated or ready to go. So there's a lot of intentional mining, tidying, curation that needs to go when you pull down this information from all of these different sources, both public and proprietary, so that it is apples to apples and your model can actually learn from it. But there's so much of that that exists out there. And that's what we call unsupervised data, right? This is just sort of like you want to expose your model to as many examples as possible so we can start playing little childlike games over and over trillions of times. Think of it like fill in the blank exercises, right? Where, if I say the girl kicked,
Mhm.
you have learned that probably something's going to come after that. It could be the ball, it could be her brother, right? But there's a narrower universe of things that could come in that fill in the blank than if you just started a sentence with the. And
Yeah.
so, in the same way with these protein sequences, the model, as it's exposed to it, it's learning the language, it's learning the syntax and grammar, it's learning what protein domains are responsible for what functions, it's seeing evolutionary dead ends where things didn't work. And ultimately, what it comes out with is the ability to write things from scratch. So I'll be very clear here. Our models are not starting with a existing sequence and then performing the AI version of targeted mutagenesis. They are being fed input where you say, I have this therapeutic problem. To
Mhm.
solve it, I need an enzyme with these qualities. And it says, okay, let's start with amino acid one. If I know what I want the end protein to be capable of doing, what should the next amino acid be? What should the next amino acid be? And it generates millions of these possibilities and then ranks them based on its own prediction of how confident it is that that's going to be a successful hit. And that's where the second type of data comes into play. So you have this unsupervised natural data that you're exposing the model to train it, right?
Mhm.
Now you have supervised lab data. So there's this sort of like feed forward loop where if our models have generated certain sequences and the model thinks that these are going to be, CRISPR analogs, for example, right? Now you have to go test them, right? So you take the top five and maybe a couple sprinkled around that it doesn't think are going to be as good, and you put them into the wet lab and you run functional assays and you see how they perform in real life. And now you've gone from like the theoretical, the hypothetical, it's like rubber meets the road. Like, what does this actually look like in practice? And when you get those results back, that's supervised data, it's labeled data. So you feed that data, positive or negative, back into the model. And it learns which of its things were successful, which ones weren't. And maybe it sees patterns in terms of why, what made the successes a success, and vice versa, right? And so, it then learns from that data. And I think that's a really interesting point that ties into sort of, the idea around both is there enough data and what kinds of data are there? I think, in the field of biotech and medicine, there is an emphasis on successful data. That's what
Mhm.
gets you papers. That's what gets you drugs, right?
Yeah.
Publish or perish. And yet in the field of tech, there is a very different attitude towards quote unquote failure. It's, move fast, break things, learn from them, right?
Mhm.
And that view of when something fails, you can learn from it. The models will learn something. So it's not a waste of time. It is, in fact, if your models were only exposed to positive data, that
Yeah.
would not be a good thing. They would not be as well rounded. And so that for me was a very interesting psychological shift that you're almost, as intrigued by negative results as you are by positive results.
Yeah, and I've heard anecdotally as far back like as you can imagine, at JP Morgan last January, AI was there was a lot of buzz about AI, and I sat in on multiple discussions with folks who talked about that drought, I guess, of bad data, and that being like potentially a pretty serious problem to the industry's embrace of this technology. Now, and I want to ask you a question about that. Like, you're so convincing, Hilary. Like, I'm listening to you, and I'm like, why would anyone be skeptical? Can you answer that question for me? Why is there so much skepticism?
Yeah, I know I'm happy to. And I probably would say that I might count myself, or I would have counted myself. I certainly had heard about AI. It was creeping into some of the conferences that I was going to in some of my former companies, but I don't think I didn't understand it. I think that's the root of a lot of skepticism. Like whenever
Mhm.
you see something new come around, often there's a lot of top level messaging or miscommunication around what something is and what it isn't that can lead to a lot of this confusion or skepticism. So, first off, I would say it depends on the exact question that you're trying to answer. So if you're talking about skeptics who just think that AI in general and or for biologics is just total hype and not actually reality, you know, I would say, like, is AI a magic wand that you can just wave at a difficult therapeutic problem that your best team's been trying to solve for 20 years and it just instantly solved? No, it's not. So if that's the type of hype we're talking about, then I would probably agree, right? Um, is AI a highly complementary tool that can be used to augment the pace of innovation in science and in therapeutics? I think yes. And the key that I see there to it having a realistic impact, and it is having that now, to be clear, is collaboration. It's not some machine learning scientist like locked away in their cubicle coding and then coming out and saying, aha, I've solved, cancer. It's this tight collaboration of folks on the AI machine learning side working with biologists, manufacturers, clinicians in whatever the biological vertical is, and saying, look, you know the problem. You have intimate knowledge of how to design a therapy, what a clinical trial looks like. Tell me things. Tell me what your ideal attributes are, and I will translate that into what the model needs to be exposed to and what types of output that it's spitting out. And when you have that tight collaboration,
Yeah.
particularly when you're talking, in our case about CRISPR and AI, those are two, hyped up, sexy, inflammatory topics, right? But then disruption plus disruption, where's that going to take us? I think that's phenomenal. Maybe building on the second point, then you have folks who think that it's powerful but don't really understand how it works. And so then have that sort of level of skepticism. Maybe it's not skepticism. Maybe it's just not understanding how to apply it, right?
Yeah.
Excited, but don't know how to apply it. And I think my executive coach would say, reframe your skepticism as curiosity, right?
Yeah.
It's really a two-way street here. And I don't know where the burden lies, but I would say there's some responsibility for leaders in AI to educate both society, but specifically the broader, scientific community about exactly what AI is and isn't, how it
Well,
can.
I think the burden, where the burden lies probably has yet to mature to the point of
Yes,
burden, but I
yes.
think it's going to come real quick, right? Like
Oh, very quick.
the burden's going to fall on drug developers when they see other drug developers developing drugs
It
using it.
absolutely, it
Right.
absolutely is. And to not just like brush it off as sci-fi, but like, on the other side of the coin, on the developer side, there does also need to be that spark or that curiosity around, I want to learn more about this technology. I'm interested in how it could help me. It's one of those things where when somebody tells you that you can just go boop, boop, boop on a computer and you're going to get some, perfect proteins, potentially perfect proteins spat out. There's that innate sort of like, oh, hell no. Like, you know what I
Mhm.
mean? I've been doing this for years and I have spent,
Yeah.
time, blood, sweat trying to make these therapeutics. How is it possible that like a robot can do this, right?
Right.
And then you see it in action. And if you're able to get over that kind of sense of like jealousy or, sort of like, how is this possible? This invalidates everything I've been working on personally, right? And see the promise of where that could be taken with, tight collaboration and supervision and guidance, you start to see, oh my goodness, we could take some of the most knotty problems that folks have been like, because what is the ultimate goal? The ultimate goal is not how you get there. It's
Mhm.
just to get there. It's to have the medicine. And if AI can help with that, I don't see why we should treat it any differently than any other tool that we have brought into the toolkit over the past decades of pharma.
Yeah. I want to double back real quick on something we were talking about earlier that I think relates to that skepticism. And how are you doing on time, by the way, Hilary?
Oh, I'm fine. I can keep going.
Good, because we could talk all afternoon. I want to double back to the point you made about validation in the wet lab. I think we were talking about data, and you were talking about like, you cycle these outputs back to the wet lab, and then that wet lab experience creates more data, perhaps negative, to get back to that negative data thing, perhaps negative, and you feed that positive and negative data back into the machine, this iterative process. And I want to one make sure I'm correct here. ProFluent is conducting that wet lab work, correct? Like,
Yes, yes, we are. So we have both the dry side of the lab, which is more the machine learning AI side, and then we have a burgeoning wet lab so that we can carry out some of that initial validation ourselves. Yes.
yeah. So am I right also in assuming that the work that goes on in the wet lab, the outputs of that work get fed back in, like I said, positive and negative data, and
Correct.
therefore contribute to the learn, right? To the learning.
They do. I mean, that's part of, I think, how it got the name generative AI, because it's this idea of generations or iterations. So, the model will generate and create these novel from scratch sequences. We go into the wet lab, we make them, we test them out, generate data. That data goes back into the model. It takes a look at what worked, what didn't, and it then iterates on those parent sequences and creates child sequences that usually are better, right? Because
Mhm.
it's learned valuable things. And that iteration process back and forth, most of the problems that we've worked on to date, we've needed about 2 to 3 cycles back and forth. And so it's incredibly fast
Mhm.
to be
Yeah.
able to get sort of that validation and then immediately have better options to try out.
Yeah. All right. Now I'm going to fast forward again, bounce back to the skepticism thing, and I don't want to beat this horse. Like I don't want to overstate the skepticism thing. But from my perspective, I mean, I cover the space. I talk to a lot of folks, and I see it all the time. But I'm curious about your perspective as a business development person. Like, you're the business development person who's like out there engaging the industry with this solution set that ProFluent is working on. What are you seeing in terms of industry appetite?
Yeah, so perhaps a couple of musings here. One is that I think we are seeing a trend where if you look back 5, 7 years, most of the talk around AI in pharma was more around better analysis of clinical trial data, patient
Mhm.
data,
Mhm.
or could it be used to, streamline manufacturing processes, quality control, stuff like that. And what you're seeing more at JP Morgan and many other venues over the past 12 months has been, does AI have a place in earlier stage in discovery and development research, and a lot of examples that are proving out, yes, it fundamentally does, across a wide array of verticals, right? I think some of the first comers were more in the antibody biologic space, and then you see folks doing small molecule research with AI and then other types of proteins. And so, I mean, it's here. And I think, in terms of the industry appetite and adoption for it, that appetite's definitely there. From my perspective, on the BD side of the startup, there's a distinct bifurcation between sort of like flavors of big pharma. So on the one side, I see certain companies that have really invested enough
Mhm.
to know AI. And it's not always folks being like, I'm going to go get a second PhD in machine learning, but they bring in the right people to sort of augment their capabilities. And those are the folks who can do deep diligence, right? Because nowadays there are hundreds of companies who plaster sort of AI, machine learning, deep learning on their website. When you look under the hood, you get a sense of, how cutting edge and innovative are they in the field of AI. When something's new and people don't know much about it, the glitz and the glam can be very easy, right? To sort of
Right.
hide things. But like, is a company using, say, somebody else's open source code and recycling it for their own purpose? Or have they made AI advancements in and of themselves? What do they have platform innovation on the side of AI, not just on the side of drug development, right? And so that's kind of like you have, that sort of flavor of big pharma that's made the investment, that understands it, and that knows where they want to apply it. These are
Hmm.
the folks that'll come to us saying, we have a problem. We have strategic alignment or focus on a particular indication. We see this particular mutation in this patient group's DNA, and none of the tools that we're working with right now can efficiently address that mutation, fix that mutation. And we know that to address it, we need something with these exact criteria. Can you build that for us? Right? That's a conversation that's fun. Then perhaps the second bucket is more the folks that see that AI is sexy. They see that everybody's talking about it, but they don't understand it. They have some fundamental skepticism, or they want sort of, a proof of concept, a look-see, a pilot. They want to test it out before they invest too heavily. And
Hmm.
often they don't know what they want to use it for. And, our CEO, Ali, is fond of saying, why bother using AI for problems that could be solved without AI? Reserve the big guns for the problems that are intricate and complicated enough that you can't solve them by yourself, right?
Hmm.
So, like, we don't want proof of concept experiments. We want big, meaty, ambitious moonshots, right? Because
Yeah.
that's where you really test your technology out, right? That's where, you figure out the difference between hype and reality.
How do you sell that though? I mean, how do you like you're a BD person who, you're heading up BD for this company that, as I said, is doing some things open source, giving some stuff away. You want these great big giant projects, these big problems to solve. How do you like I'm curious about give me a little hint as to what the business model looks like there right now.
Yeah, so I think, we are super early stage right now.
Hmm.
I've been here for 6 months. And so I think, ultimately strategy is always going to be a moving target that needs to evolve, with the changing times and the changing markets. I think we looked at sort of the early stages of the company and said, what is our strength right now? Um, we're small, 25 people roundabouts. What we know is protein design. What we have is some amazing machine learning scientists and some amazing editing scientists who really can make that feed forward loop go quickly. And
Hmm.
so why not play to our strengths? And why not have a success before you sort of build too ambitiously, right? Why not, Ali's paper from back when he was at Salesforce in Nature Biotech showing the first example of somebody could take large language models and write proteins from scratch, and then they worked, right? That was a moonshot in and of itself.
Hmm.
And to go from the theoretical to the practice, and then to start thinking of, how are you going to apply this, right? Why not find partners who are the experts in that given field or that given disease, who understand what it takes to take something from that early discovery lead stage and get it to the clinic on sort of that accelerated timeline. And we each play to our strengths, right? A lot of, this is maybe more in sort of like the biotech or the startup world, but I will say that often what happens is when you're fighting for VC money and fighting to stay alive in markets like this, one of the first things that often gets cut is your platform team. Because often by then you have a couple of programs in the clinic or heading to the clinic,
So
and
let
you just
game
can't
be on
just
those.
yeah, you can't justify keeping a platform team alive if all you're seeing is sort of, they're putting in tons of hard, amazing work, but they're getting kind of these incremental advances in proteins. And so in some ways, that's, what we are in a module is, your protein design team from your wildest dreams, right? With capabilities that, again, a lot of companies haven't necessarily invested in the AI infrastructure to be able to do protein design at the level that we can do it. And so, as we've been, as we debuted, one of the reasons I think for open sourcing Open CRISPR was this idea of how do you prove that you have what it takes? to live up to some of these promises, right?
Mhm.
And to be able to show that, for the first challenge that we took on publicly, right, wasn't necessarily a let's go find some like small, easy enzyme family and then see if we can make, a competitor to that. No. Ali looked for hard problems. I remember some of our earliest conversations, and he said, "Tell me about editing. Tell me about white space. How easy is it to make an editing therapy?" Which got everybody to laugh because it is so hard
Yeah.
build something that is safe and efficacious for patients. And he said, okay, let's go. Let's build something here that works. And so, you know, Open CRISPR was not us trying to build a SpyCas9 competitor or some perfect editor for a given use case. It was literally a proof of concept of us saying, we've trained up a model, we've fine-tuned that model. If we now let it run and tell it to make something similar to Cas9, but with a completely dissimilar sequence, can it do that? And do those sequences function? Cutting DNA is not an easy task. And going through that project and coming out with some top hits that had amazing activity, that was absolutely phenomenal, right? And so Open CRISPR itself, that's just one sequence out of millions in our portfolio. So in terms of business value, the inherent value around one sequence, it's not quite the same. Like I think if you look back at sort of the earlier days, there's a lot sort of focus on static assets, on, well, I own this molecule, I own this sequence, right? And I will license it to other people who want to make medicines using that molecule. I think we're maybe moving a little bit beyond that to less of, what the sequence is, but more of, can I build the best thing that will fix this problem? And there are maybe hundreds of ways I could go about building different things to fix that problem, right?
Mhm.
And so then it becomes a little bit more around sort of those ambitious problems and the products that you're helping somebody design and build and test. And so we just ultimately made the decision that what we would like Open CRISPR to achieve is accelerating innovation. There are a lot of folks out there who are very interested in solving problems with CRISPR technology, but for a variety of reasons, maybe they're kind of like, a little intimidated by the intellectual property landscape. Maybe they can't afford the royalties. They're looking for other options. And so you don't see open sourcing really happen in the world of biotech. It's very common in tech, right? Where it's like, new software, I'm going to just give you my raw source code and you're going to break it and you're going to make it better. And the whole idea behind that is to advance the pace of innovation. Right. And so, we started wondering similarly. I thought it was a crazy idea at first. I'm not going to lie,
Mhm.
but it's grown on me. This idea of, that was never going to be per se our moneymaker. There are tons of enzymes out there that are simple cutters, that are nucleases. And so why not just give this one out to the community and say, use it for whatever you want. Use it for research, use it to build a therapeutic. You don't owe us a cent. Just go and make good things with it. And, what was fascinating to me is I kind of thought, what kind of demand are we going to see? What kind of interest are we going to see? And, from a business development perspective, I think when I look at the past for given sequences, depending on your licensing paradigm, exclusive, non-exclusive, maybe you see 4 or 5 players bidding on something. We got hundreds of license requests in the
Yeah.
first week, which we were totally unprepared to deal with, frankly, right? That type of demand we just hadn't anticipated. And from such diverse sources, folks in agriculture, folks in biomanufacturing, folks in therapeutics, big pharma, academia, small startups, right? And it sparked a lot of really interesting conversations that led beyond Open CRISPR and more to, okay, if this is the proof of concept of what your models can build, now build me this other thing that will solve this other problem I have. And so, stay tuned. I think you're going to see some interesting things from ProFluent in the next year.
It sounds like it. And not the equation, the recipe, like what you're putting together there, new as it may be, and agile as the company may be, right? Like the direction is not necessarily carved out in stone, but you're doing well. I mean, you've attracted some pretty significant investor dollars. I mean, I think I read somewhere like 44 million.
Yes, we have 44 million in funding.
Yeah. So I mean, you're in good shape there, and we're going to stay tuned. And I would love to continue talking to you all afternoon. We do need to wrap it up though. We're going way long, but we'll do a part two. How's that? We'll
Good.
get back together. There's so much more to talk about, but I want to wrap things up a little bit with like, you know, maybe we can't get too crystal clear on what's happening next with ProFluent. But you said from the outset of this conversation, I remember you told me when we first spoke a couple of weeks ago, because I remember responding to you. You said, I didn't know what I wanted to be when I grew up. And I think I responded to you and said, I'm 49 years old and I still don't know what I want to be when I grow up. A lot of people are hoping that I don't choose to be a podcast host. But you've been studying, like we talked about it, like you got into the gene editing space, you stayed in that therapeutic area for a while, and then obviously made this move to ProFluent, which is a bit of a departure. But I'm just curious, like what sort of has driven, beyond the things that we talked about, what have some of the drivers been that have led you to the point where you are now, and what do you see coming down the pike? Like what's the next sort of pivot for?
I mean, I wish I knew. I think, it's definitely fair to say that I am not the kind of person who has a 5-year plan. That being said, in retrospect, I think I can pull certain patterns from some of my pivots and moves. And I think, one of them is definitely the technology, that I'm attracted to sort of disruptive, new, risky technology, because I think that that's ultimately where we're going to see some of the biggest payoffs.
Mhm.
And it's just so closely connected to my mission of seeing other patients with rare disease get solutions. Um, I, but even more so than the science, because again, I mean, around the Bay Area, around Cambridge and Boston, you spit and you can find good science, right? There's a lot of good science, a lot of good technologies. I think the other major factor has always been the people, that you sink, in some ways more of your life, more of the hours in your day to interacting with other people at your workplace than you might do with some of your family some of the time.
Mhm.
And you want those to be good people, that you want to be in a room at 2 a.m. in the morning trying to figure out that next step in drug development or get that deal done that's going to lead to the progression of a program. And that has been a constant theme. I have made, you don't have to be best friends with everybody that you work with, but I have made some lifelong friends at Editas, at Vor, at Tome, and now at ProFluent. And that is going to I think be a continuing theme, working with other like-minded people who are hardworking, who are intelligent and competent, and who are just decent human beings that are in it not for the glory, not for the VC dollars, not for the potential ROI, but really, really just want to help people. And this is the way that they've chosen. And I would imagine that that's going to be where I continue to center interest around opportunities. And at some point, I might decide to take the plunge and kind of go full force into patient advocacy. Time will sort of tell. Like I can't wait. I think I should like do one of those like message in a bottle to myself. Like if it had been 10 years ago, I never would have said this is where I would be sitting. And yet here I am enjoying every second of it. Right. So like I can't wait to see what the next
Yeah.
10 years bring.
Well, you'll be continuing to do the Venn diagram exercise
I
as
will.
part of sort of yeah. Your enthusiasm and excitement, and knowledge is it makes it very clear, right? Like that you're in the right place right now. And I've learned a lot in this conversation, and I'm glad that you came on to spend some time with you. And I am also disappointed because I would like to spend the next 2 hours continuing the conversation, but I just can't do that. So
Anytime.
yeah, we'll definitely do it again, but I hope you've enjoyed it as well. And I'll be in touch to get a round round two scheduled.
Sounds good. Thank you so much, Matt.
Thank you. So that's ProFluent Chief Business Officer, Dr. Hilary Eaton. I'm Matt Pillar. You just listened to the Business of Biotech. If you want to hear some different perspectives on AI and biotech, earlier this month, our own Tyler Menichello hosted a bioprocess online live panel discussion exploring how the tech is being applied in process development and manufacturing. That is available now on demand under the Listen Watch tab at bioprocessonline.com. So go check that out. Come on back for a fresh episode of the Business of Biotech on Monday. And in the meantime, thanks for listening.